All right, so good afternoon, everyone. Hello, welcome to the HNRCA Monday Seminar Series. We're excited to have you join us today. And if you're joining for the first time, welcome. Thank you for being here. The Q&A box in Zoom is open. And if you have any questions for the speaker, you're welcome to submit them at any time during the talk. We'll have a Q&A session at the end of the seminar after the presentation. Our speaker today is Dr. David Wishart, Distinguished University Professor in the Departments of Biological Sciences and Computing Science at the University of Alberta. Dr. Wishart has extensive experience in metabolomics and bioinformatics. He's the co-director of the Metabolomics Innovation Center and has made major contributions to both the Human Metabolome Project and the Human Metabolome Database. And here's where it gets even more interesting. Recently, Dr. Wishart has been involved in characterizing chemical constituents in foods and food-associated biomarkers. So our audience will be especially interested in hearing about these applications of your work to nutrition and food science. We're so excited to hear your talk today. Without further ado, I'm now pleased to introduce our speaker, Dr. David Wishart.
Thanks very much, Catherine. It's a pleasure to be here virtually. I was actually in Boston just a couple days ago, and I was tempted to stay on a little bit longer, but I have to teach here this week, so I'm doing it remotely. So as advertised, I'm going to be talking about the application of metabolomics to nutrition and food science. Hopefully everyone can hear me okay and can see the slides all right. This is a brief outline, so I'm going to try and give everyone a bit of a background around metabolomics, and then I'm going to show you how metabolomics can be applied to a number of different areas. One in the area of food composition analysis, and another area in terms of dietary assessment. And then particularly interesting to me and perhaps to the field now is the application of metabolomics to understanding diet and the microbiome and how it relates to health. And then I'll close off with a very brief discussion about the application of metabolomics, machine learning, and precision nutrition. So in general terms, metabolomics is defined as the study or characterization of a complete collection of small molecules found in a given biological sample. So that sample could be a cell or tissue or biofluid or an entire organism. And when we talk about small molecules, we're talking about things that are generally having a molecular weight of less than about 1,500 Daltons. So these are not proteins. These are things like amino acids and nucleic acids and organic acids and biogenic amines. The approach to metabolomics is and I think the appeal is that it's a way of rapidly and inexpensively and comprehensively and quantitatively characterizing not only metabolites, things that we think of traditionally as constituents in metabolism, but also to identify things like the essential nutrients, which may include vitamins, and non-essential nutrients that might include phytochemicals and polyphenols, or non-nutrients, a variety of natural products, food additives, drugs, and supplements. So basically, metabolomics is broadened to cover just about any kind of organic molecule that can be found in a biological system. In the case of humans, there are literally hundreds of thousands of molecules that are in us and in our food. One way of thinking about metabolomics is this diagram I often show, which is something I call the pyramid of life. At the base of that pyramid is the genome. So that's the DNA that's in every cell. The genome codes for the proteome. And the proteome is there really to help manipulate and catalyze and transport the metabolome or metabolites. So we use genomics to study the genome, proteomics to study the proteome, and metabolomics to study the metabolome. As you climb up the pyramid, there's an interesting trend. The influence of environment and physiology grows as you go up the pyramid. So the genome is inert. What you eat or drink or breathe doesn't change your genome, but what you eat, drink, or breathe does change your metabolome, and sometimes on the order of seconds or minutes. Likewise, the genome in your heart and the genome in your brain and the genome in your skin is no different. But the metabolome in your heart, your brain, and the skin is profoundly different. So this is why physiology and environment play a real close role or important role in the metabolome. And so technically the metabolome is the closest chemical interface or omics interface to the environment. And so probing the metabolome allows us to understand not only gene, but environment interactions, and therefore to explore the phenotype. So this is why it's become kind of a hot area over the last number of years. When we're measuring metabolome, we can start with something like some tissue samples, biopsies. If they're solid tissues, we have to do extractions. But in the case of studies with mammals and humans, we usually work with biofluids first. So we can work with urine or saliva or blood. And then we go through a variety of chemical analyses. And I've pictured some LC, HPLC, mass spec, NMR. You can use gas chromatography, Fourier transform infrared, all kinds of techniques. These techniques have been around for a long, long time, but what's happened over the last 10 or 15 years is the development of software, which allows us to go from the instruments to the step that I've shown on the left, which is the data analysis. And thanks to computers, databases, and software, we're now able to analyze really complicated mixtures and measure not just 1, 2, or a dozen compounds at a time, but measure literally hundreds or even thousands at a time. And so this has really revolutionized a lot of analytical chemistry and opened the door for some really interesting applications. Now, different types of instruments that we use in metabolomics have different levels of sensitivity. Nuclear magnetic resonance is the least sensitive. It has a lower limit of detection of around maybe 5 micromolar. GCMS, low micromolar, high nanomolar range. And then liquid chromatography, mass spec, capillary electrophoresis, tandem mass spectrometry. These are much more sensitive. They can measure things in the sub-nanomolar range. A number of compounds you might see with NMR, numbers around 100, GCMS, maybe a few hundred, LCMS that could be measured in terms of thousands or even tens of thousands. The challenge is that with greater sensitivity, we end up seeing a lot of stuff that we just don't know what it is. Whereas the less sensitive tools like NMR or GCMS, we know mostly what they are. So those are called the knowns, and then there's the unknowns. And this is still a continuing challenge in metabolomics, but that's the subject for a completely different talk. Now in metabolomics, there are at least 2 routes that are used, or 2 methods. One method is called targeted metabolomics. Another approach is called untargeted metabolomics. The more common approach is untargeted, and it's essentially where you use a very high resolution mass spectrometer typically, and you do lots of what are called chemometric analyses to align and cluster peaks and identify interesting features. And from the starting set of maybe 10 or 20,000 features, you winnow it down to maybe 100 or so things that are interesting or different, and those are ones that you'll try and identify. Targeted approach is probably more relevant, more useful for analysis of foods and nutrition and food science. In this case, it's using a pre-selected set of compounds where you have isotopic standards or reference standards of some form, and you can use mass spectrometry or even NMR spectroscopy. In this method, you're actually quantifying and all of the expected compounds in your system or your kit. Targeted metabolomics allows you to, in many cases these days, identify and quantify more compounds than untargeted methods. And some of the newer methods give you up to 1,000 different compounds. My own preference is for targeted, and some of the results I'll be presenting will largely focus on the targeted techniques for metabolomics. A lot of the information that has been assembled over the last 15 or 20 years in metabolomics is in this data resource called the Human Metabolome Database, or HMDB. We've been building it since 2006, and we're now getting into 16 years of maintaining it. It's a really comprehensive database, and it's more like an encyclopedia. It contains a lot of information about all the known human metabolites, about 250,000, their structures, their pathways, where they're coming from, concentrations in different biofluids, the functions of metabolites in terms of their biological roles. There's a lot of reference spectra, mass spectra, NMR spectra that helps people identify compounds. So the numbers are big, millions of spectra, hundreds of thousands of pathways, information on hundreds of diseases. It captures information both from the targeted and untargeted metabolomics data, but the most useful data certainly is the targeted metabolomics data because it gives us real numbers that we can work with. So in compiling and maintaining this data resource for the last 15 or 20 years, we've learned a few things. We've learned that humans eat other metabolomes, so that our metabolome is made up of many different metabolomes, from fish to fruit. And so a lot of that comes from our diet. A lot of it comes from exposures, inadvertent or deliberate. So there's the endogenous metabolome that our bodies produce through known metabolism. There's processed metabolites that come through digestion and microbial metabolism. And so that represents sort of the food metabolome, which I think is of most interest to the folks here. But then there's also drug and pollutants and their transformation products. So we've learned that the human metabolome is not just simply the endogenous collection of things that our cells produce, but the exogenous collection of many hundreds, thousands of exposures and tens to hundreds of thousands of chemicals, most of which we don't know, but a large portion of which are in this database called the Human Metabolome Database. So I'm going to dive in a little bit and talk about how metabolomics can be used for food composition analysis. Just remembering that most foods come from living organisms, and so metabolomics is used to characterize what's inside living organisms. So extending metabolomics from the human to a fruit or an animal or even a processed food product isn't very hard to do at all. So if we look at the traditional idea of food composition analysis, which has been around for probably more than 100 years, early days, it was careful titrations, bomb calorimeters. People use UV spec and GCMS now to help characterize things like total fat, cholesterol, sodium, total carbohydrates, dietary fiber, protein content, sort of the macronutrients. And then more recently, over the years, people have been adding more information on some of the micronutrients like vitamins and certain minerals. But typically what we characterize or think of as in terms of food composition analysis is what we see in the USDA database. So the nutrient database is huge, covers almost 250,000 food types, both prepared and raw. The composition data typically ranges from about 77 compounds or components up to 90 nutrients that are listed in the food composition database. The USDA database is the oldest one in the world. It's the most comprehensive one. It's used and borrowed by many other countries that have their own country-specific databases. And this is the perception we have generally of what is in food, somewhere between, let's just say, about 20 to 30 components. But food is much more complicated. And given that we know that there's 250,000 compounds in humans, we should expect about the same number of compounds in beef or veal or pork or even fish. And there may even be more compounds in fruits and vegetables. So clearly we're just scratching the surface of what's in our food. So when you apply metabolomics for food composition analysis, it's a little different than traditional approaches. So we'll take food samples, we'll extract them, then we'll run them through our mass specs and our NMR, and we'll use many of the same databases, tools, software tools to identify those compounds in these different food types. So with metabolomics, instead of just thinking about a dozen or 24 compounds, we're thinking of hundreds or thousands of compounds, and we're thinking of using a whole range of high-end analytical instruments. So what are the advantages of metabolomics? So as I mentioned before, metabolomics allows this rapid quantitative measurement of hundreds to thousands of compounds. It's much faster than many of the traditional methods. It's often with targeted methods to analyze an entire sample in about 10 minutes. It's really cheap, about $40 per sample, and it's obviously more comprehensive. So those are some of the central advantages, and this is one of the reasons why people are moving towards metabolomics for food composition analysis. My own lab has been developing kits or systems for doing targeted food analysis. These look at a variety of lipids and lipid-like molecules that you'll find in samples. They also look at organic acids, amino acids, various dipeptides, amino acid derivatives, nucleic acids, biogenic amines, other types of compounds. Those are sort of broken down into hydrophobic and hydrophilic molecules. These kits are always changing and always evolving as we get more information and as more needs are requested and met with our development team. But these are, consist of plates and solutions and standards that are all put into a box and then fired into a mass spectrometer. So we've used kits like these, as well as other methods to characterize a number of different foods. A couple of years ago, we characterized the milk metabolome, or the milk composition database. So this is a combination of both experimental work as well as literature work. So in the milk you may have had this morning in your cereal, there are at least 2,300 compounds that are within a typical glass of milk. We have a large number of concentration values. We have a lot of reference spectra. All of the compounds are described in detail. And like many of our databases, it supports a lot of structural, spectral, and text searches. So this is an application of what metabolomics can do to characterize a food product. Typically, if you look at the label for milk, you might see maybe half a dozen compounds, but in reality, it's thousands. We've also worked with other groups around the world to help characterize polyphenol content, and we've developed and released this resource called Phenol Explorer. It's now in version 3, and it contains information not only on the polyphenols, but also the transformation products from polyphenols, content in foods. It's collected from peer-reviewed papers and also from studies we've done. There's about 500 polyphenols found in about 500 different plant foods. We also look at polyphenol metabolites and the effects of food processing on polyphenols. We've also been compiling a lot of the data that we've measured, other labs have measured, and obviously what's in the literature to create a food composition or food constituent database called FooDB. This hasn't been formally published, but it's being widely used by a lot of people right now. It's about 71,000 compounds in about 800 different raw or fundamental foods, as the USDA calls them. And we have information about things like their flavor or aroma, color, their effects on human health. We have lots of concentration data, lots of references. Not every compound has a health effect, not every compound has a flavor or aroma attribute, but a lot of them do. And again, like our other databases that are online, it's freely available and people can search through things like structure or spectra or text to find out more about these things. It's going through a bit of an upgrade, and we hope to be able to publish officially FooDB sometime early next year. Another initiative that actually grew from our work on the FooDB was inspired the Rockefeller Foundation and I guess the American Heart Foundation to lead the Periodic Table of Foods initiative. This is a 4 or 5 year project that started in late 2021, early 2022, to characterize using metabolomics the composition of about 1,000 different foods. They're recruiting a wide variety of labs from around the world, and they're building out metabolomics kits, similar to ones that I've described, to help the different labs characterize these food constituents in a quantitative and consistent way. The net result will be a resource not unlike FooDB, which again will allow people to explore not only composition of foods, but also look at regional differences in how is an apple from Canada different from an apple in the US versus one in England. So these are examples, I think, of the applications, the new types of information that we're able to generate, and the new initiatives that are arising from advances in metabolomics towards food composition analysis. The next thing I'm going to do is switch to using metabolomics to characterize our diet. This is a little different than characterizing food. Now, traditionally, when nutritionists, food scientists are thinking about characterizing diets. They'll do things like food frequency questionnaires or 24-hour recalls. Some cases they can do weighed food records. They can take food diaries and histories. Some cases people will be housed in metabolic research kitchens and everything is monitored. Those are obviously much more expensive than food frequency questionnaires. But there are problems, and I think people are quite aware of these ones. So there's issues like recall bias or incorrect recall. People are really lousy at portion size estimation. There are effects that come from both reporting and observing. There tends to be problems with underreporting. And people have noticed this in particular with obese subjects, where for a long time it always seemed that obese people seemed to eat less than people who were of normal weight. And it wasn't really sorted out until we realized that there was quite a bit of underreporting. Obviously, if you're trying to do complete characterization, if you're doing a metabolic research kitchen, cost, staffing, duration are all limited. Typically, what we found is that standard FFQs and others are good for calorie intake and broad macronutrient intake estimation, but not for getting exact consumption, not for understanding the micronutrient intake, or understanding the effects of nutrient absorption or metabolism. So what people have been moving more and more towards is the idea of finding measurable, relatively non-invasive biomarkers to assess both macro and micronutrient intake. So there are things like double-labeled water, which is the best method for quantifying total intake. There are things like measuring plasma ascorbate to understand vitamin C intake, looking at carotene as a marker for vegetable consumption, there's compliance biomarkers to look to see if people are breaking the rules or drinking things like ethanol. But more recently, and an initiative I was involved in for the last 6 or 7 years, is the idea of developing biomarkers of food intake, or BFIs. And these can be both quantitative or qualitative. And the idea is to identify these and use them as a way of monitoring what people are actually eating. So in terms of dietary assessment, metabolomics does allow us to start looking at these BFIs, as well as the concentration, replacement, and compliance markers, because we're able to measure so many different chemicals. BFIs are these food-specific metabolites. They have to be basically unique to the food. They have to be measurable. They have to be ideally quantitatively correlated to the consumption of a specific food. Now, some of these compounds are quite rare or unusual. So metabolomics is actually pretty good at doing these unusual compound measurements. One of the better known BFIs, and maybe the inspiration for metabolomics to be applied to dietary assessment, was a compound called proline betaine. This is something that was published about 10 or 12 years ago, looking at the correlation between proline betaine and citrus consumption, and how it has this remarkable specificity and sensitivity to identify if and when people have been eating citrus fruits. So this represents almost a perfect biomarker for citrus consumption in terms of its performance. There are other kinds of BFIs. Some are more specific, some are less specific. So typically if people drink tea or coffee, that's one of the main sources of uric acid, which is found in the urine. Meat, methylhistidine, red meat, I think it's 3-methylhistidine. Eggs and other lecithin-containing products will produce trimethylamine oxide. Wine or beer, ethyl glucuronide, soy, equal. Onions will produce hydroxyphenyl acetate, and so on. So all of these are compounds, the ones in red, are examples that are potential BFIs. Some are highly specific, some aren't so. But the idea is that if we measure these compounds, we have a pretty good idea of what people are eating or have been eating over the last 12 to 24 hours. So it's because of that information and these discussions and papers that were appearing that something called the Food Biomarker Alliance was launched. And this is a joint European Union initiative that also brought in New Zealand and Canada and other participants from other countries. It ran for about 6 years. And the concept was to use metabolomics, but also to scan the literature to help identify and characterize the food composition biomarkers and BFIs. And from that, we were to try and create things like Metabolomics kits, some of which I've just shown you, and databases, which could be distributed and shared around the world. So when we measure BFIs, we take food samples, but then we give them to people. So it's not going straight from food to sample. And so from people, we will then take urine and biofluids like blood or saliva, and then measure them through the standard metabolomic pipeline. So in this case, we're analyzing biotransformation products from the food that we eat. Now, sometimes things are transformed, like the example of hippuric acid. Others, like proline betaine, aren't transformed. And typically the non-transformed metabolites are, it turns out, more useful. So we've been developing a dietary biomarker kit as part of this effort with FooDB. This one measures about 120 compounds. It measures a number of metals and metal ions, short chain, medium chain, long chain fatty acids, a variety of phenolic acids that represent polyphenol breakdown products, a lot of vitamins and cofactors. And so these cover markers of meat and fruit and vegetable consumption. We're expanding this. This kit will be somewhere over 200 compounds by, I hope, the end of December. But again, it's always changing, always evolving. In addition, this consortium, which probably included about 70 or 80 different scientists, did a lot of literature reviews to assemble information. And some of these things were also confirmed through studies with metabolomics to look at biomarkers for meat intake, tuber intake, onion, tropical fruits, seaweed, apples, pears, bananas. And a lot of really interesting and useful biomarkers have been identified. And some of these are now being migrated into various metabolomic assays. They've also, the information from these research activities has been put into online databases. One is called PhytoHub, which is maintained in France, and we've been working with them for a number of years. This now has about 1,700 food phytochemicals, their metabolites, about 1,200, and it links these components to almost 400 different types of plants, fruits, and vegetables. It's designed in the same model or just similar layout to our other databases. In addition, we've also been putting this information into a database called MarkerDB. And this one is sort of a general biomarker database. So it has genetic markers and karyotype markers and protein markers, but it also has more than 1,000 metabolite and dietary biomarkers or chemical biomarkers. And so these can be used to assess food exposure. And we've tried to capture information about sensitivity and specificity and ROC curves. A lot of this is captured from the literature and captured from the papers that we put together as part of the FooDB project. And then it's expanding because more and more work is being published in this area. So whether it's PhytoHub or MarkerDB, these are, I think, really useful resources for tracking what's going on in terms of biomarkers of dietary consumption. I'm going to jump now to how metabolomics can be used to look at diet, the microbiome, and also relate that to health. So certainly through Human Microbiome Project, study of metagenomics, we've learned quite a bit. And one thing that we certainly have learned is that the diet, the microbiome, and the metabolome are really closely linked. And that we should really look at microbes as basically as chemical factories. They're there to supplement digestion and supplement the transformation of foods. In some cases, also to produce important essential nutrients for our bodies, or to convert a lot of phytochemicals that we can't normally process into something that can be. I showed this diagram earlier about the different food biomarkers, how hippuric acid or TMAO represents a marker for egg or tea consumption. But I've also colored here how these compounds are transformed and how particularly the red ones represent microbial products. And so about half of the ones that I'm listing here represent either microbial or microbial human co-metabolites that lead to the markers that we see. So, this is just a random sample. We get the fact that about half of them have microbes touching them at some point, tells us just how important the microbiome is to not understanding food biomarkers, but also for the production of important micronutrients. This is another example where we're just taking, if you bite an apple or bite into an apple, you'll typically produce a polyphenol. It's called isoquercetin. That'll be in your mouth, but immediately it's deglycosylated to go to quercetin. Quercetin will be absorbed in your bloodstream, and it'll go either to the liver, where it'll produce a number of other phase 1 and phase 2 metabolites, or it could go straight into your gut, where it'll be transformed through microbes into a variety of other compounds, where there'll be various ring cleavages and breakdown and production to some compounds that are helpful and some compounds that are actually harmful and dangerous. And these are things that I think we're just starting to understand and realize, that even though an apple a day is supposed to keep the doctor away, some of the compounds that we think are supposed to be healthy may not be, or there's a mix of both good and bad that come from them. In the case of microbial derived metabolites, many of them actually form what we call uremic toxins. So if you know a little bit or recall a little bit about uremia, now this is about damage to the kidney, but it turns out the uremic toxins also can damage the heart, the bone, vasculature, liver, brain. And that many of these things relate to microbial metabolites and the interaction of our dietary chemicals with those microbes. And particularly if there is disruption or gut dysbiosis, a leaky gut, a lot of these compounds end up in our bloodstream, and those can cause pretty serious damage. These are essentially endotoxins that are produced from the food we eat. So I think there's been a real revelation over the last 5 or 10 years where we've started to understand the connection between the gut and the brain, the gut and the kidney, the gut and the lung, the gut and the liver. And these are mediated not just by the bacteria, but by the chemicals they produce. So in the world of microbiomics, everyone talks about good bacteria and bad bacteria, but it really is more about good chemicals or bad chemicals produced by good bacteria or bad bacteria. And in that regard, there are metabolites that both heal and harm their host. So here are some examples of the harmful metabolites: cresyl sulfate, indoxyl sulfate, deoxycholic acid. These are all produced by microbes, often Clostridia microbes. Some are associated with chronic kidney disease, behavioral disorders, neurodegeneration, oxidative stress, anxiety, neuroinflammation, depression. So these are things that come from our diet, but are transformed by microbes, and which lead to long-term chronic disorders. There are other things which come from the fiber in our diet. And again, it's the microbes that convert essentially indigestible fiber into these helpful organic acids like butyric acid, which is a remarkable anti-amyloid, HDAC inhibitor. Acetic acid also is anti-inflammatory. And there are other metabolites that come from ellagic acid, which also exhibit anti-neuroinflammatory effects. So these are compounds, some of which we know, others which are maybe new to you, which have also been shown to have beneficial effects. And I've put these down, and these are more focused on brain health, but the list is growing in terms of many other areas, from heart, cardiac health, liver health, and gut health. And the ones in green are the helpful ones, and the ones in red are the bad ones. Some of them I've already mentioned, like butyric acid, acetic acid, but there's also things like indole propionic acid. Others like lipopolysaccharide, TMAO, creosote sulfate and doxyl sulfate all seem to have roles in the development of dementia, Parkinson's, Alzheimer's, autism. So these are things that clearly have an effect, and they play very much in terms of what we eat, when we eat, and what our body happens to do with them. An interesting story that emerged from metabolomics, it's now about 10 or 11 years old, is the uncovering of the connection between microbial metabolites, diet, and cardiovascular disease. So this was done by Stan Hazen at the Cleveland Clinic. And basically what they found was that there was a compound called trimethylamine oxide, which was associated with people at high risk for heart disease, stroke, and it was always elevated. And what they found through a number of years of research was it was very much associated with diet. So people who eat fatty foods, we do know that they tend to have higher risks of cardiovascular disease. And fatty foods have phosphatidylcholine. Phosphatidylcholine is converted in the gut to choline. The gut microflora will then convert the choline to something called trimethylamine. That trimethylamine will go to the liver and it's converted to trimethylamine oxide. And trimethylamine oxide is an atherogen, or it causes atherosclerosis by acting on foam cells. And so the idea is, what can we do to try and reduce the risk and reduce TMAO? And again, Stan Hazen's group looked at some of the things, interventions that, reducing fat and meat intake. They also tried to look at enzymes that would prevent conversion of TMA to TMAO. But they also eventually found that the most useful target was targeting the bacteria, and a particular enzyme called trimethylamine lyase. Trimethylamine lyase is found in firmicutes. It's not found in all microbes, but it specifically converts choline to trimethylamine. Turns out that some people have lots of these firmicutes in their gut, and so the net result is they, even if almost on a fat-free diet, they'll still produce huge amounts of TMAO. Well, there are other people who have almost no firmicutes, and they can eat all the cake and bacon they want, and they never develop this TMAO. What they found was that there is actually a compound in olive oil and also in red wine, grapes, and red wine vinegar called 3,3-dimethylbutanol, which looks a lot like choline, and it inhibits trimethylamine lyase. And so it turns out that people that have diets high in olive oil and wine and grapes, i.e. the Mediterranean diet, are generally able to kind of wipe out the activities of the firmicutes and the TMA lyase. And this might explain one of the reasons or the rationale why the Mediterranean diet reduces heart disease. But also other discoveries that are emerging, not only in the area of neurodegeneration and heart disease, but also metabolic syndrome, again, identifying metabolites, again, coming from food and microbes that will either help or hinder the development of metabolic syndrome. So again, we see short-chain fatty acids, we see butyric acid, we see trimethylamine, we see cresol. We also see a new compound called imidazole propionic acid, which also seems to cause things like insulin resistance. So these are examples of well-known and even new compounds arising from our food and arising from our diet that lead to physiological effects. What we've tried to do over the last couple of years is assemble that information into an open source database called the Microbial Metabolome database, or MindDB. And this is a collection of all those microbial metabolites that I've mentioned and many, many more, their origins, their food origins, the reactions, the enzymes that mediate them, and their effects on human health. So the database has almost 25,000 metabolites now, from about almost 2,000 microbial species that are found in humans. And it links these things to hundreds of different food sources, as well as hundreds of different human diseases with descriptions of their mechanisms and associations. Database is trying to bring together many sources, not only metabolism, but also microbes and food information and information about the biospecimens, the health effects, and bioactivity. So this is actually probably our most ambitious and complicated database because it tries to bring metagenomics, genomics, metabolomics, exposomics, all those omics together into one resource to help inform us about what's known and what's being discovered. And it's intended not just as essentially a data resource, but also potentially a discovery resource. So for the last bit here, I'm going to talk a little bit about metabolomics, machine learning, and precision nutrition. So it's very brief, but it's partly just to highlight this idea that with metabolomics, we're generating lots and lots of data. We call it big data. Also with microbiomics, we're also generating huge amounts of molecular data. Now, the concept behind precision nutrition is to help customize an individual's diet using this big data, this molecular data, to optimize health and wellness. Now, a lot of these precision nutrition studies are being based on large cohort, multi-omic, genomic, proteomic, metabolomic studies. And again, this is generating huge amounts of data. So whether it's the omic data or the multi-omic data or the large cohort data, we're dealing with huge amounts, terabytes of data. And that overwhelms the traditional statistical methods and many of the scientific methods that we are used to. But we do know, and I think many of you are hearing about artificial intelligence and machine learning, and these are computing techniques that allow us to see patterns in the big data, to start making sense of it. And this is, I think, where all of this is sort of leading to, which is, we have the tools to generate these things cheaply and quickly, or generate data. We have people who are engaged and interested in their health, but can we make something useful out of that? So the concept, again, is to take some of the metabolomics tests that I've described, as well as the other genomic tests, and to measure them on or perform them on people. And then to take that data and to use data analysis, use the data resources that I've talked about, to use machine learning tools, the visualization tools, to help interpret that data, and then to come up with actionable information on nutrition or exercise or supplements, or even modifying it for medications. Now, there are a number of companies that are picking up on this. Some of you may have heard of Day 2. There's places like Derived, Molecular U, the Great Plains Laboratory, which has been around for about 20 years and maybe was one of the first to do this. Habit. Some of these companies appear and disappear, but I think most of them are still all around. But they use things like targeted metabolomics, just like what I described. And they'll often use microbiome tests, and they'll use those to try and identify either nutritional abnormalities, diet propensities that people may have. But then they'll also use their own databases as well as machine learning to help customize diets to address either nutritional deficits they see or the excesses, or to modify their diet to make people a little more responsive to certain types of food or nutritional interventions. Many of these companies also use life coaches, and they also use continuous monitoring, where people might be providing blood or urine samples on a weekly or monthly basis. So does it work? I think there's some really interesting case examples, especially with Day 2, where there are some really interesting diet combinations that were identified that seem to have some positive effects on individuals, especially controlling their glycemic index. It is clear that it requires constant monitoring and quite a bit of data collection. And these are not purely metabolomics methods. Now there's a lot of wearable devices that they use, and these help find optimum diets, optimum foods, and customize things for best results. The effects are limited. They're best in the areas of pre-diabetes. They seem to have some applications in mitigating memory loss and sort of slowing down or even reversing some general aging. There's a major initiative coming from all of us. It's being sponsored by the NIH to look more into precision nutrition and collecting multi-omics data. So I'd say that really this is at an early stage. It's probably a bit more hype than fact. But I think the progress is happening, and I think the tools are there. So with that, I'll wrap up. I know we're running a little behind on time, but I just wanted to thank my collaborators as part of this food ball project, some of my staff, and colleagues who help develop the data resources and our funding agencies who help make this all possible. So thank you very much.
Thank you, Dr. Wishart, for your excellent presentation. That was really impressive. It was so nice to see you describe the landscape of all these nutrient and food related metabolites. So let's go into the question and answer section.
Sure.
I'll start. I can read you questions that have come in. So first question, this is from Larry Parnell. Can you comment briefly on approaches to assess the presence or even quantify minerals in food?
So some of the best ways to measure minerals are using inductively coupled plasma mass spec, or ICP-MS. We use that a lot to measure mostly metals. There are obviously other methods to measure anions like phosphate and chloride. And so those often use some form of electrochemistry. The ICP-MS seems to be really popular in terms of both its reproducibility, precision, and sensitivity. So that's, I think, one of the better methods.
Okay, great. This question is from Ala Mohammed and Al-Fudeh. Are there any identified known sets of biomarkers or metabolites that are increased or decreased with certain diets, such as Mediterranean diet or high fat or high protein?
Yes, there are. I'm not sure if I could give a list immediately of some of the ones, but certainly high protein diets, we tend to see much higher levels of things like creatinine in urine. We'll also see higher levels of methylhistidine, 1-methylhistidine and 3-methylhistidine, depending on the source of the protein. High fat, we'll often see perturbations to certain lipids, lipid classes, saturated fatty acids, which will be elevated a fair bit. TMAO, another marker typically of high fat diets. In terms of, there are sort of secondary markers that will sort of show up as effects of the diets. So I think high fructose consumption can generally increase the uric acid levels. Some of the markers that I showed in my presentation, where there are certain foods that are associated with them, you can kind of pull them together and say, okay, based on the what I know is typically in a Mediterranean diet or what's in a high fat diet, you'll see these or associate them. The marker DB has some of that information in terms of the things that are associated with certain foods and the specific markers that are elevated in blood or urine.
Yeah, and there's a related question about dietary patterns or foods that can be carcinogenic, such as charred grilled meats. Do you know anything about the food composition for those?
Yeah, I mean, certainly the polyaromatic hydrocarbons are produced when we char or burn meat. And some of the Maillard reaction products also may have some potentially carcinogenic properties. The other thing that's probably less appreciated is that many of the uremic toxins that I've mentioned also are carcinogenic. And when they're present in the body for long periods of time, which is what happens when we eat with a standard diet for the same period of time for years or decades, that also increases the risk for cancer. So there are ones that are known to directly cause cancer, like the polyaromatic hydrocarbons, but then there are these others that are more the microbial products, which cause mutations over sort of a longer period of time. I wrote about this in a review article, I think that came out earlier this year, about multi-omics view of cancer, and talks about some of the dietary effects and exposure effects that lead to carcinogenesis.
Okay, great. Let's keep going. So the next question is from Michael Lustgarten. Thanks, David, for all your work in metabolomics, especially the HMDB. His question is, well, TMAO negatively impacts cardiovascular outcomes in animal models. It's also found in fish, which is associated with cardiovascular health. Do you have any thoughts about this disparity? Is the TMAO story one of poor kidney function, like you can't properly excrete it instead?
Yeah, it's something that's been, I think, debated a little bit. I think there are, clear direct effects in TMAO, and they've been shown in lab animals and in cell systems. The TMAO is elevated in cold water fish. Fish that are pulled out from north of 50 or 60 degrees latitude. So TMAO is very low in a lot of sort of Mediterranean fish. The other thing is that TMAO that's in fish goes straight into the gut and straight out. So it's not as elevated in the blood, and it's sort of temporarily elevated. What's happening with the bacterially produced TMAO is it's sort of this constant source of it. And so, just eating anything that has lipids and anything that has protein in it will still produce the TMAO. I know there was a real pushback when the TMAO story first emerged, but I think that the feeling now is that, yeah, the TMAO effect is real, and that the TMAO linked with fish is historically just with very cold water fish, and that the TMAO is pretty much eliminated when it's because it passes right into the urine.
Okay, our next question is from Christopher Wiley. Flavonoids like quercetin or fisetin are rapidly sulfated or glucuronidated and excreted into the bile. Is there evidence for secondary processing of these conjugates into harmful compounds by gut bacteria?
There's kind of a moving back and forth that goes on with things. So, once things end up in the bile, they end up in the large intestine, where the microbes start working on them. So then they'll deglucuronate and desulfate things, and then they'll break open the rings and convert them into something else that's a bit more soluble. And then that passes back from the gut into the bloodstream again, where you'll see some other phase 1 or phase 2 or more microbial metabolism. So to go from quercetin all the way to hippuric acid, there's something like 8 different steps and at least 3 different body compartments that are involved. So it's biotransformation isn't quite as simple where instantly things go through one step and it's gone. There is this multi-compartmental transfer that seems to be going on, much more so than we probably realized.
Okay, a question from Alan Taylor. Why do you characterize pterodeoxycholate as beneficial versus deoxycholate as toxic? What is the difference between their physiologic effects?
That's a good one, and I'm not sure if I'd be able to give a really good answer for that one. I think that part of it is the modification and enhanced solubility. With bile acids, there's really some striking differences where just a single change from a taurine or a sulfate or a glycination of the bile acid will change it from harmful to helpful. Some seem to play a role in enhancing the permeability of the blood-brain barrier. Others seem to actually reduce the permeability. I think what you're kind of dealing with is they're steroid drugs, and steroid drugs can vary tremendously in terms of their effects just from small modifications. And I think that has more to do with either steroid receptors or some of the multi-drug transporters about what they do to them. So it's probably a hard to answer question that has a lot to do with protein drug interactions.
Okay, thank you. For BFIs, what patient factors are the most important that will influence the magnitude or explain variation in BFIs?
Well, I think that the best BFIs that people are finding are the ones that are not transformed. That is, they're not converted in people's gut to something else or converted through some general metabolism. So proline betaine is a great biomarker because it exists in the fruit and it passes straight through the body without any modifications. Hippuric acid is not a great biomarker for any specific food because it's the end product of many, many different transformations from many different polyphenols and probably even some from food additives. You know, equal is a good marker for soy because it's specifically produced by microbes. There's not a whole lot of other transformation products, but not everyone has the microbes, and so you'll have a slightly diminished amount for different people. So I'd say that microbial metabolism is maybe the biggest influencer of these BFIs, which ones are good and which ones aren't so good.
Thank you. So I'm looking at the time. I think this will be our last question. So this is from Shangdong Wang. It's a little bit long, but I think I'm going to try to simplify it. So he says it's very interesting, the use of metabolomics for studying food metabolite associations. I was wondering how food metabolite associations could be replicated or validated by metabolomics, because there's a lot of factors which influence food metabolite associations. And he mentions a few of these: sample preparation, cooking methods, metabolism of hosts, etc., all making them very challenging to validate a particular metabolite as a dietary biomarker. Could you comment?
Yeah, and I think those are all good points that limit the efficacy of these BFIs. And so, as I say, the best BFIs are the ones that are not transformed. So proline betaine doesn't get modified with cooking. Proline betaine doesn't get modified by microbes. Proline betaine doesn't get modified by phase 1 or phase 2 metabolism. And so there's not as many compounds as we hoped that are inert to transformation, either from cooking preparation or from our gut physiology or our own physiology. But given that any given food has about 100,000 different chemicals, the odds are you are going to find something that is inert to transformation. And so the markers that are being tracked by Foodball, the ones that we're trying to put into both the papers and the biomarker databases, generally seem to pass those quality controls. I think the main issue with food biomarkers is that they are temporally limited. So, if you ate a banana 6 months ago and haven't had a banana since, we won't know that you ate the banana 6 months ago because anything that it produced has long been eliminated from your body. So if we wanted to be able to measure things over longer term, we might need to look at adipose tissue or hair or fingernails. And there are some interesting markers that I think we can see that allow us to look at compounds in hair or fingernails that can measure things over much longer periods of time.
Okay, so this is going to conclude our Q&A and today's seminar. Apologies to those whose questions we did not get to, but we've run out of time. Thank you to our speaker today. Thanks to all the attendees for coming. We'll see you at the next HNRCA seminar in 1 week. Thank you.
Thanks very much. Bye for now.